Hello, everyone. My name is DeMarcus Turner, and I am a NOAA CCME graduate scholar studying at Florida A&M University. And so I am in my second year getting my graduate degree. I'm seeking my Master's of Science degree in environmental science, and my area of focus within CCME is in coastal intelligence. And so I have the pleasure of doing this amazing project, which is validating a model of microplastic hotspots from Apalachicola watershed to the Apalachicola Bay. And so here I am out in the field, at least trying my best to look like I'm busy. And so we're going to get into just this amazing world of microplastics and a lot of the information that we are trying to gain from them. And before I start, microplastics is exactly how it sounded. Just a year ago, I had no clue what microplastics were. Just kind of heard the word thrown around in science circles, and it's exactly how it sounds. They are microscopic plastics, plastic pieces that you can only see under the microscope. So anytime I do research, I find it very important to get back to the history, the culture, the background of the area of focus, whether it be from a broader scope and kind of narrowing our horizons. And that's what you'll notice, particularly in this presentation of this research. So just to go over a brief history of pollution generation, it starts in the middle of the 19th century with the Industrial Revolution, where the world was introduced to industrial and agricultural and medical advancements that preserve human health and human life, which of course was the aim and which was the focus. But it wasn't until the middle of the 20th century that the effects of pollution were felt, and the world was also introduced to the water and air pollution. So in the 1960s, lawmakers made conservative efforts to put forth legislation and awareness. And so by the 1970s, we had legislation like the Clean Air Act of 1970, followed by the Clean Water Act of 1972. Coastal and marine pollution is largely connected to waste discharge from land masses, and also discharges from human activity on the sea and ocean, whether it be wreckage from ships, whether it be debris from nets and equipment and things of that nature. And so 80% of debris in marine environments originate from land-based sources. And of this debris, plastic material is the most prevalent macro pollutant in the world's beaches and oceans. And it's important to note that plastics make up 60 to 90% of all marine debris studied. And I was kind of reminded in one of my classes today, particularly in ecology, about these great Pacific garbage patches. These giant garbage patches, the most well-known is the one in the Pacific, just off the coast of California. But for what we know right now, there are 5 distributed in the major gyres throughout the world. So, and I bring that up to say that it is projected and studied right now that all this trash, all these patches, and all the debris around in the seas and ocean, 60 to 90% of it is projected to be plastic. So this is a brief history of plastic. It was first created by Edward Simon in 1839, who invented the first plastic, which is polymerized mixture of synthetic organic compounds. And Alexander Parks followed Edward in 1862, the first man-made plastic called Parkesine. And then you have Leo Hendrik Baekeland in 1907 with the first modern plastic bakelite, which is the first plastic that is commercially trademarked, which is the plastic that we are most familiar with in today's time. So these inventions followed with mass production of plastics in the 1940s. And so I want to kind of bring up Edward Simon again, because in 1839 and Alexander Parks in 1862, because this is around the time of the Industrial Revolution. This is a lot of different advancements were being made in the industrial industry, in the agricultural industry. And so, I just kind of wanted to bring that up because this is kind of what it kind of stems from that era. But of course, you have a situation in the 1940s and the 1950s, where pollution is just starting to get studied and just starting to really get observed. And so in the 1950s, for example, plastic production was only approximately 1.5 million tons. And in 2016, it had reached 335 million tons. So now we kind of narrow our scope to talking about pollution in Florida. There's very little information on microplastic concentration as it relates to Florida. The Florida coast is, but what we do know is that the Florida coast is lined with sources of microplastic pollution due to industrialization, urbanization, and agriculture. Plastics are circulated through storm water runoff, wastewater treatment plant effluent, tire wear, and atmospheric deposition. A recent study done, McEachern and others estimated that Tampa Bay contains approximately 4 billion particles of microplastics. And I took this particular study out of my research, not only because it's recent, but because it's a good way to compare and contrast my area of study and also an area of study in two different parts of Florida. In the area that I'm studying, the Apalachicola River and Bay, you have an area that is, very well protected by the federal government. It has a lot of different research reserves. It has a lot of care that's taken to it, different types of strategies and plans. And then you take the Tampa Bay area, which is a very urbanized, industrial, agricultural area, highly populated. And I really wanted to compare and contrast the two different environments to see what are we seeing differently in Apalachicola Bay as it relates to the Tampa Bay area. Because the simple fact that one area is strictly a Bay area, and then you have the Apalachicola River that flows into the Bay Area, and you have different hydrological systems going on. But that's just something that will be good for the discussion section and not pertinent in the research as of right now. So this is the Apalachicola River Basin. The Apalachicola River Basin is part of the larger Apalachicola-Chattahoochee Flint River system. The Apalachicola River is the largest river according to flow and is the most active estuary in Florida and flows 107 miles directly into the Apalachicola Bay estuary. It assists a profitable commercial and recreational fishing industry, and the ACF river system is connected to Georgia and Alabama in addition to Florida, and its increased urbanization has resulted with increased population growth, water resource demand, and point and non-point source pollution. And microplastics fall into the non-point source pollution because we can't track exactly where these plastics are coming from, but we know that they are typically coming from stormwater runoff and runoff from the coast in the coastal areas that are neighboring it. So this map that you see here to the left is actually a model that I will be validating. It was designed and created by a member of my committee, Dr. Rebecca De Jesus, who is an associate professor at Louisiana State University. And this basically shows the microplastics that are emitted into the water, ACF watershed in kilograms per day, with the light colors being little to no microplastics and the darker colors being a high amount of microplastic concentration. And so this model is strictly based on GIS software and hasn't really been validated by field methods, which is my job coming into this. And these green dots, and I find it important to note, we'll talk more about it later. These two dots are dams, and this dam up at the top is the dam that's really going to be a focus and play a key role in my research as well. So there are different ways to categorize microplastics: by size, by color, by structure, and even chemically. So plastics are categorized into three different size classes. You have microplastics, which is 1 or less than 5 millimeters. You have mesoplastics, which are 5 to less than 25 milliliters. And you have macroplastics, which are greater than 25 millimeters. And so the category that I'll be focusing on in my research, of course, are microplastics. And the microplastics have varying sizes and very in different shapes. And there even is a subdivision in microplastics where you could have nanoplastics, you can even have picoplastics, depending on the size. And so these aren't example of microplastics per se, but just so you get a understanding of the shape that we're looking at under the microscope. And I'll show you some microplastics under the microscope and what they really look like according to scale. But some different structures that you have example of are things like the one at the top left, filaments. To the top right, you have jagged edge pieces. In the bottom left, you have these films. And then at the bottom right, you have styrofoams. So the size of microplastics make it consumable to marine fauna as small as plankton. And I find it comfortable saying that now because of where I am in my research. I'm still in my data collection phase when I'm actually looking at the microplastics and getting different sizes. And the smallest that I found was a length of 40 nanometers, which is very small. So I'm very comfortable in saying that these microplastics have the potential to be consumed by marine fauna as small as plankton. And its impact can influence food webs from the bottom up. The biggest topic right now in studying microplastics is really looking at the chemical composition. Part of which, and then I'll talk about a little bit about it later, is what I do want to incorporate eventually into my research, because the chemical composition is very key to understanding the impacts that microplastics may have on marine environments and marine ecosystems and marine life in general. Commercial and recreational wildlife like oysters and fishing inhabiting the bay are sensitive to environmental changes, particularly microplastic generation and distribution. And right now in the Apalachicola Bay, specifically for oysters, they're seeing a change in some of the salt water that is coming into these estuaries. It's becoming a little bit more saline. It's becoming a lot more saltier. It's a lot more salt water is being pushed into the estuary and into the river, and it's having a negative impact on the oysters, not just from the increased salt levels, but also from the overfishing. So they decided the government, I think even the Florida DEP decided to stop oyster fishing in most parts of the Apalachicola Bay and Apalachicola River. So I'm going to go over the purpose and objectives of this study. Presently in the ACF Basin, there's little known about microplastic abundance, accumulation, distribution, and variety. So the purpose of this study is to validate a model that tracks the generation of microplastics reaching the Apalachicola Bay. The first objective is to assess the presence or absence of microplastics in sediment and water samples. Basically, to see if we see any microplastics, whether they're there or whether they are not. And then the second objective is to determine the density of microplastics in sediment and water samples. Kind of looking at to see what is the most abundant structure. Are there more filaments? Are there more films? Are there more styrofoams? How big are they? And how have you. So now I'm going to go over the field methods that we are using for this particular study. So the first sample site starts just before the Jim Woodruff Dam, which is right here, the top green spot here. That is the Jim Woodruff Dam. And we sample just before the Jim Woodruff Dam, which is located, it's like a border between Georgia and Florida. So further upstream is the Georgia side, and then the further downstream you go is the Florida side. And the rest of the sites are spaced out until just before the Apalachicola Bay. And the reason why we decided to just do the Apalachicola River and not continue into the Apalachicola Bay, because we really wanted to see how the flow of the river and the different types of sediments within the river and the different factors at hand really affects the density of microplastics. And of course, we want to look and focus on the hotspots shown by the model. So sediments were collected from the center of the river as best as we can. There was only one site in particular where we had to go a little bit closer to shore and away from the middle of the channel, because the bottom of the riverbed was just so hard and rocky, and it was really tough to use our Pono grappler, which you see in these pictures here. So this is the Ponar graph sampler that we used, or the Ponar, and how this particular mechanism works, if you're not familiar with it, is this is in its closed state, in its closed position, but in its open, you have these openings when you do prepare it for launch that appear at the bottom. And then there's a pin, you can kind of see the pin right here, that you put into the holes when they connect. And it has a spring on its end, and you have to put it in just right so the mechanism works. So you tie the Ponar to a rope like the yellow one you see here. You guide it into the bottom of the riverbed. You have to guide it. You can't just kind of throw it overboard because the pin might fire prematurely and then it'll close. And of course, in many cases that I experienced in the field, you won't catch anything but water. And water is nice, but not when you're trying to get sediment. And so after it reaches the bottom, you kind of jerk it. You don't want to pull it. You want to just jerk it just to activate the spring. When you activate the spring, it closes like you see in this picture. And it, in this case, anyway, you successfully catch some sediments. And here we got some very muddy sediments. And we'll talk about why that's very interesting as well later. So after pulling this up, we collect sediments in 250 milliliter jars and store them at or below 20 degrees Celsius. In this picture, you can see that this isn't a glass jar. This, you might say, hey, DeMarcus, that looks a lot like plastic. It is. It's called a Nalgene container. And we use only Nalgene containers for our sub-sediment samples that we were going to use for each site to study grain size. And we'll go over that in a bit as well. So this is the method that we use to collect our water samples. This is a 330 micron plankton tow net, which is approximately 50 centimeters in diameter. For each of the sediment samples, not including the sub-sediment samples, there was a water sample collected. And so typically with a plankton tow net, you tie it to the back of the boat and you drag it along the way. We found that a different method might work just as well because we want to look at the density of microplastics in the water. And it's just easier to look at density when you are able to collect water in a receptacle that has a known volume. So we decided to grab a metal bucket, a metal bucket that I found at Walmart, submerge it under 1 liter of surface water, and then we kind of hold the plankton net on the side of the boat, and we pour it directly in. And typically with plankton tow nets, at the end you have a plastic receptacle, but of course we didn't want to do that, given the topic of discussion. And we were able to find a glass bottle around the lab that fit perfectly. And so you have a metal bucket, which is about 20 to 22 liters, and we pour that amount of water, starting from the top of the plankton tow net, let it run all the way down until it flows. All of the water flows either out of the net or all of the sample flows into the glass bottle. And we then take the glass bottle, we poured out the sample into a 250 milliliter glass jar and stored at or below 20 degrees Celsius. So for each site, we did duplicates, each for the water samples and the sediment samples. It was easy to do the Ponar graph sampler because we have 2 different chambers. We're able to collect 2 samples at once. And so here, I'm going to talk about my laboratory methods in processing these samples, starting with the sediment grain size, which I feel like is going to be a very key part of the research as well. So the grain size for each location is recorded using subsamples from each sample site. And the reason why I really feel like the sediment grain size will play a key factor is you can already kind of see it in the picture, the different types of sediments collected from different locations. These are the sandy and rocky ones, the lighter ones. You can see that they're a little bit more coarser, and the darker color ones, they are the lighter ones a little bit more finer, the darker ones are a little bit more coarser. And then you have these 3 in the middle that have a mixture of both, which you don't see in the picture because they were still incubating overnight, or the other few samples. And they were our mud samples, the ones that you see being collected here. And they needed a little bit extra care, particularly because they are mud samples. Like I had to dry them overnight, and when I came back the next day, they were like clumps of clay, hard, like they had been baking in the oven for quite a bit. And so they needed just a little bit extra care to make sure that I could loosen the sediment properly so we can get an accurate sediment grain size. And so each of these samples were wet sieved through a 63 micron mesh and then oven dried at 50 degrees Celsius. So the parts of the subsample that were sieved through the wet sieving process had a sand fraction of less than 63 microns of the subsample. And then the subsamples from that were then dry sieved. So we used, I think, 5 to 6 different dry sieves of different sizes, you know, with the largest size being at the top and the smallest being at the bottom, and then you had the catch pan right underneath. So they were shaken until all of the sediments ran through as much as they could. And then I recorded the different amounts of weights of grain sizes based on the size of mesh that they were caught in. So that part of the experiment I was able to finish, and there was just a wide variety of different grain sizes amongst the different sites. And I really feel like that this would play a key role based on if microplastics would just so happen to get to the bottom of the riverbed. You know, I believe anyway that the more sandy soils or the more rocky soils would pretty much won't catch them very well, and they would just continue to flow down the river, and that we will be able to see that. And in the case of the muddier samples here, I feel as though if the microplastics just so happen to float to the bottom, that because they're muddy, because they're, you know, goopy and mucky, that the microplastics may get caught. And it's important to note that these muddy samples, interestingly enough, were only found around the Jim Woodruff Dam. And so we believe that one of the things that we wanted to also look at is if the Jim Woodruff Dam and these muddy sediments are a potential sink for microplastics, which could be something that we might test in dams in different parts of the world just to see if that's consistent with the data. So this is the laboratory method I used to process the sediment samples. This is a density separator. It's simply made by some sealant in some PVC pipe. And this is just the overall layout and blueprint of how you to build it, the connector pieces that you would need, the clear PVC pipe, the white PVC pipe, and the vials. I was originally told that, of course, I might have to build my own density separator, and I'm not particularly handy, but I am willing to do whatever I can for science. And I was just so thankful to the people at Ander for providing this density separator that before I didn't have to, you know, go through trial and error trying to build my own. So definitely shaved off a week or two off of my preparation in that regard. So can't thank them enough. Can't thank the folks at Ander and Jason Garwood enough for that. So processing the sediment samples through the density separator takes approximately 26 minutes in entirety, not including cleanup between samples. And after they are run through, and I'll go over how exactly this works in the next slide, because there are other instruments and factors and tools that are used in addition to the PVC pipe. So the vials are after they are processed, are rinsed into sterile glass vials. And then the sediment samples in these vials, in addition to the water samples that were collected earlier, are filtered through vacuum filtration. And then the sample is stored in a sterile Petri dish. Each Petri dish is labeled with the sample number, the type, the location, and the date. So this is the density separator that I used at Ander, that they were I was very thankful that I was able to use. And there, as you can see, there are different factors at play, and I will point out each and every one as best as I can. And so, of course, here in the middle of the picture, you have the PVC pipe, and attached to it is a bucket. And in this bucket is salt water, and you kind of see the salt here to the right. And this salt water was used throughout the entire system because, of course, these samples are coming from salt water. So we wanted to simulate that, because using fresh water might mess with the densities, it might mess with the flotation of the microplastics as we're trying to process them. And so I was able to gauge around the amount of parts per million of salt water I wanted using the YSI device, again, given to me and allowing me to borrow for the folks at Anner. So the salt water was between 15 to 25 parts per million. And this power source here, this battery is powering the water pump, connected to the hose and connected to the bucket. And so this hose connects to the PVC pipe right here, and it brings the water all the way up until about halfway in this clear zone. That way we can load in our sample at the bottom, at the top. And so after the sample is loaded in, this clear tube that you see here is actually an air stone, the air stone that you typically see in aquariums and you see in fish tanks. And so this is the most important part, a part that I was very tedious on, because it is assumed that science has already been proven, as other microplastics has been studied and processed, that this air stone goes into the PVC pipe and it goes as far as down as it can, while still creating bubbles. And the theory is that the bubbles from the air stone is what brings the microplastics to the surface. And as I continue the water flow through the PVC pipe, it comes into the other end of the PVC pipe, this part right here, this opening right here. And at the bottom, when it's running directly over the bucket, is a mesh underneath that catches what is flowing out at the top of the PVC pipe. And that's where we are collecting the sample and potentially the microplastics, of course. And so here is an example of microplastics seen under the microscope, and you get a chance to see exactly how small these microplastics are. And you have the different examples of what they look like, and there are a wide variety of different colors that they may be. Here in A, you have a green filament, and B, you have a black jagged piece, and in C, you have an example of a film, and in D, you have an example of a pellet. And a pellet can come from a variety of places, typically from hair care products. That's typically how you identify a pellet. They're typically very odd, shiny, and smooth. And yeah, so these are what we are observing, of course. And one of the things that I found challenging is really trying to distinguish, particularly with filaments, is the difference between organic matter and the microplastics. And there are so many different methods. One being that you just kind of heat the tip of a pen or a needle, and you kind of touch it. And if it kind of like folds or kind of shrinks, then it's plastic. If it doesn't, it's an organic matter. And it's something that I was kind of hesitant at first, because we want to save these microplastics and hopefully get them tested for chemical composition. And kind of putting them or even potentially burning them might affect that analyzed process. So basically, we kind of just use best judgment. Is it shiny? Does it have color? And how have you? And we allow the machine that analyzes the chemical composition of the microplastics to do its job, because it will tell us very accurately if it's organic or if it's a microplastic. So these are my hypotheses or expected findings within this particular project. So if the GIS model is accurate, then there should be variability between sampling sites, whether it be upstream or downstream. And in the map I've shown you earlier, you have some darker spots, which is, again, very interesting because it's before the Jim Woodruff Dam. This is where it's projected to have the highest concentration of microplastics. And you have different light blotches. This one is a little less concentrated, a little bit more concentrated in the middle of the Apalachicola River, and pretty high in concentration depending on where you are as you go down all the way into the Apalachicola Bay. Okay. And so also, if the ACF dam is a sink for microplastics, then the concentration of microplastics upstream will be more than downstream. And so we took it upon ourselves to sample 2 different sites before the Jim Woodruff Dam and right immediately after the Jim Woodruff Dam, just to see if there is a variation, if there's a difference is there really something there as we predicted that the Jim Woodrow Dam might be acting as a sink for microplastics, and even the sediments around the Jim Woodrow Dam as a potential site for microplastics. Another expected finding is that if sediment grain sizes vary among samples, then there will be variation in microplastic sizes as well. And so, like I showed you earlier, just the different types of types and the different textures of sand that was seen of sediment all throughout the Apalachicola River. You had the muddy, coarse sediment, you had some of the rocky sand, and then you had some finer sand. And we really believe that there will be a variation of microplastics as it relates to that as well. So this is the Jim Woodrow Dam. This is a picture that I took while we were out in the field. Very interesting sight to see. I don't know why, but I'm always impressed to see just these dams, man-made structures. And we really feel like that this is going to be a very interesting aspect to add in our study. And looking forward to see if there are any other projects that might have done a similar study. And if not, maybe even be the first. And I also thought it was important to note some notable structures along the Apalachicola Bay as well. This location to the left is a little bit further downstream of the Apalachicola River. And this is a notable structure because you can't really see it too well because of my positioning, but to the left of this picture, there was some construction going on. You know, you might have some plastic in the equipment that are being used, and they might leave plastic cones and things overnight. So if I see like an increase in the plastic, I might kind of take note of that and be like, well, there was a construction site and people were working, and debris gets kind of put everywhere, and it kind of could get washed into the sea because there's a boat ramp right next to it as well. Also, I found that this is a old refinery of some sorts that has been, I think decommissioned or something. And I found this very interesting because when I was studying the subsediment samples, we found oil. So that was something that we thought was interesting and important to note, just because it was kind of cool to see. Well, kind of cool in the sense of science, but not so cool because it's in the ecosystem and in the environment. And of course, we don't want any water if we can in our environments. Notable structure that we saw along the Apalachicola River are bridges. I've seen quite a few bridges. There was one bridge I didn't include in particular because of the conditions while we were sampling, but this particular bridge is just after the Jim Woodrow Dam, which you can see in the background there. The reason why I wanted to note this is because one thing that people don't think about, particularly bridges that go over marine environments, is tire wear. And there's quite a bit of research studying tire wear. Matter of fact, I have a colleague of mine, Nigel, who is studying tire wear microplastics in different marine environments as well. And so being that there's a bridge here, there's a potential for plastics from tires to get into the river. And so that's something that I would take into account as I'm doing my microplastic counts, and noticing different, like if I see some black jagged edge pieces, I could make the prediction that, oh, this might be a microplastic that came from tire wear because this location in particular was close to this bridge. And so that's just something that I found it important to note as well, in addition to. And so thank you for listening to me and listening to me about this project. I enjoy this project so much. There's so many people that I have to thank. First and foremost, my faculty advisor, Dr. Martinez Colon, who is the one who assigned me to this project, and has been very helpful in helping me to shape the scientist that I am this far in my graduate study. Also, Dr. Mori, who not only is my CCME mentor, he's also the captain who took us out in the two of my sampling days. I also want to thank the folks at ANR. I cannot thank them enough for not only allowing me to use their equipment, but helping me complete my sampling on my last day because Dr. Martinez Colon and Dr. Mori couldn't attend, and they were just very helpful in helping me get through this project. The only reason why I don't have a picture of them is because it was storming, raining, and the conditions were not favorable, and we were more focused on trying to get in and out of the Apalachicola River. I also want to thank NOAA and CCME for the funding this project and for funding my education as well. And so just very thankful for you all for listening to this project. And now at that time, if anybody has anything, I will accept any questions or any comments.
All right, great. Thank you so much, DeMarcus. We do have one currently in the question box, and we urge anybody who has some other questions to drop a question in the box. Georgia Ackerman wants to know, how many total test sites do you have?
I have 12 sites in total. There are 2 just before the Jim Woodruff Dam, and then the rest of the 10 go all the way just before the Apalachicola Bay.
Oh, all right. And then we have a different question. Do you consider physical attributes in the river like bends? Are microplastics found in concentration by bends in the river?
Are they found in concentration? It would depend. That's not typically what my focus is on this type of research. Something that I would definitely consider based on the different locations. When I picked sampling sites, I wanted to go just if there was a convergence, I wanted to go just before, because along the Apalachicola Bay, along the Apalachicola River, there are plenty of creeks. And so, but specifically for this study, we focused on the hotspots that were shown in that particular model. And so, that'll be something that I take into account, as I look at my sites again and look at my coordinates, if there was a bend, or was there more here because there's a more straight and narrow path, or was it a little bit slower here because there was a bend, so that's something that I'll definitely consider.
Okay, great. And we do have somebody who is wondering, what's a good way to get in touch with you about further questions?
The best way to get in touch with me, I would say using my email, because I'm always checking my email, not typically not because I want to, because I have to. So you can contact me at DeMark, DEMARK, Turner@gmail.com. You can contact me. I'm pretty sure you can find me on LinkedIn under DeMarcus Turner. So yes, those are the best ways to reach me.
Okay, great. Thank you so much. And I believe that we're posting that up on the chat just so everybody has a good way to see it. Okay, we got another question that came in from John Gifford. Has there been any effort to implement buffers along our rivers and streams, given that microplastics wash into waterways from storm runoff and hard surfaces?
Yes, it's definitely something that's still, microplastics is a very new research, so it's still being studied. And it's hard to, put something in like a buffer, because it's pollution that is a non-point source pollution, and it's not regulated. And so that's something that still has to be studied. We're still in the stages where we are studying the impact of microplastics. And that's why studying the chemical composition of microplastics is important, because it will give us an insight on how toxic or how potentially toxic these microplastics are. And so another part that I want to include in my research is studying the chemical compositions of microplastics. And I have an opportunity to, within my program, by taking, doing an internship in some different NOAA labs, working with Dr. Wayne McPhee at the NOAA lab in Charleston, South Carolina, and working with Dr. Ashok Deshpande in the NOAA lab in Sandy Hook, New Jersey. And so they are looking at microplastics in marine fauna. They are looking at microplastics in the stomachs of pygmy sperm whales. And so Dr. Wayne McPhee is doing the dissections of their beach whales, if anybody's concerned. So they are whales that have already passed on, and we are just using their body to study if there are microplastics or not. So the samples that are collected in the stomachs of the whales are taken to the NOAA lab in Sandy Hook. And there, Dr. Deshpande is looking at the chemical compositions. We have a partnership now between these two labs and between CCME, where I will use my research and I will take my samples to the Sandy Hook lab and observe the chemical composition as well. And so that's the goal, because we are seeing that microplastics is a very big issue. But the problem is that it's a fairly new and a fairly newly observed issue. So that's what I would like to do. I would like to find out what is the best way, whether a buffer or whether a type of device or whether regulation or policy needs to be put into place in order to reduce the concentration in the increasing density of microplastics. So I hope that answered your question. That was a pretty lengthy one.
No, that was perfect. Thank you so much. All right. Well, I think that's all the questions that we have currently. I'm going to turn it back over to Anita.
Thanks, Josh. And thank you so much, DeMarcus, for your presentation. I was going to ask you, are you involved with the Apalachicola Bay Systems Initiative? Are you doing any of the studies with Dr. Sandra Brook?
No, I am not. I am affiliated with Ander through Jason Garwood. He's actually a member of my committee for my thesis, and everyone there has been so accommodated. I'm so thankful to you, Anita, for giving me the opportunity. You kind of met me in passing and kind of told me in the middle of 5 seconds, hey, you should do this presentation. And of course, I said sure, because you asked so nicely.
Well, thanks. Well, we appreciate you sharing your science with us, and I have put his email, DeMarcus's email, in the chat if you'd like to get in touch with him. And want to thank everybody for coming. Thank you, Josh, for the tech assistance. And please join us next month on September 22nd. We will be actually live 5 p.m. At the East Point Brewery. And we'll have Dr. Sandra Brook there and talking about the Apalachicola Bay Systems Initiative, a little bit update on what they've been doing over the last 8 months, some of the experiments they've been conducting, and how you can stay in touch with the project. So please join us there. If things get bad and we're not able to meet in person, we'll go back to virtual. So thank you once again, DeMarcus, and thank you everybody for joining us. And we will have a recording of this up on our website.
All right, and thank you so much for having me.